detected uh, potential of polyphenolic extracts from Costa Rican wild ferns. As I mentioned, I work at the UNA in Costa Rica, specifically in the School of Chemistry, and I am the head of the Phytochemistry lab. So ferns are ancient plants that uh, develop in humid weather, especially in tropical areas, where they can find shelter and humidity and perfect conditions to their development. So, Costa Rica is a special place where ferns can develop properly because of those weather and climate and weather conditions. And in the past few years, those ferns have been studied and have helped to develop some uh, profitable products, like the one you see in the screen. Um, those products are aimed to treat some skin disorders or disorder problems like vitiligo, psoriasis, atopic dermatitis, and they have been proved to work. So, what are we trying to say here? That some products, like the one that I just showed, developed from polypodium aureum, have similar metabolic pathways to some other families like Polypodiaceae and Pterophyta ferns because they have some similar secondary metabolites like caffeic acid, bindogenic acid, and carhenic acid, among others. So, um, having that in mind, we aim to explore ferns in terms of the amount of quinic acid, chlorogenic acid, and other bioactive compounds, especially total phenolic compounds, to explore the potential application of treat skin disorders using ferns. So, what we did, we measured, well, we aimed to collect 21 wild ferns and divided those between leaves and rhizomes. We proceed to dry the samples and extract and mill the samples, and after that, we extract the samples with methanol at 75% methanol. So once we had these tracks, we quantified the quinic acid, chlorogenic acid using HPLC techniques. We also measured the antibiotic activity that those extracts might have. We use Staphylococcus aureus, Staphylococcus epidermis, Pseudomonas aeruginosa, y Escherichia coli. We use the well diffusion method and as a modification of the Kirby-Bauer protocol by duplicates. And we also did the Follensy-Caltius colorimetric method to measure the total phenolic compounds. And we did one more sample, well, one more test that was to determine the sun protector effect using the same extracts and proper dilution. And what we did was to record an UV visible spectra starting from 190 up to 800 nanometers. And once we did that, we just processed the data. So here I am presenting you the results we got. At the bottom of the y-axis, you can find the Polypodium pseudoaureum. That is the species that is being used as to produce fern block, for example. And what we found, but in leaves, here we have leaves at the left and rhizomes at the right. So we can find here some interesting point of views. Leaves have more secondary metabolites than the rhizomes. First of all, that's the first thing to notice. And second of all, that we find interesting amount of quinic acid in so many of the species that we found, both leaves and rhizomes. So that is interesting. And the range is about from, I would say that 5 milligrams per gram up to 25, 30 milligrams per gram. So it is important amount. When we move to chlorogenic acid, we have the leaves and the rhizomes as well, left and right, respectively. And once again, we have the ester on hydrolyzed at the polypodium pseudoureum at the bottom, and we found that Polypodium ursipis and Pheopeltis microclepis are higher. They have higher concentration in the struct leaves than the Polypodium pseudoreum, meaning that it could help to be used, or we could use actually these extracts to produce some interesting extracts to treat this kind of disease, aiming to that uh, goal. Um, what we found in rhizomes is that few samples had the chlorogenic acid, the whole molecule, uh, on hydrolyzed. So it is a small amount, but it is interesting as well that they have higher amounts at the polypodium pseudoreum as we collected. The other data showed that when we studied the antibiotic activity, we found that the Staphylococcus aureus have some troubles growing in the data dish, the data and the petri dish. I'm sorry, and and we have some fern extracts that showed some activity as well, similar to the Polypodium pseudoreum. And when we move to Staphylococcus epidermis, what we found is that some similar data that the Polypodium pseudoreum does have an important uh, inhibition of growth inhibition of the bacteria, but so does some of the samples as well. For example, the Serpocaulum tricelae, the first one, it has nearly 50% of the relative uh, percentage of inhibition of growth. In a similar way, uh, when we study, uh, those are rhizomes, I'm sorry, not leaves, those are rhizomes. The rhizomes, here we have uh, even more interesting results because the Polypodium pseudoreum does not have or did not show any inhibition activity, but many other samples did show that activity. So this is important to highlight because uh, rhizomes are not um, well known for these properties, and it could I could be presenting you some potential uses of this uh, plant material. So this is interesting as well. And when we move to the total phenolic compounds and solar protection factor, I'm sorry, I have one more thing to say regarding this topic. These antibiotic effects were shown only in gram-positive bacteria because the gram-negative bacteria did not show any positive results. So I have none of those results here because of that. So when we move to the concentration and solar protection, the concentration of different metabolites, such total phenolic compounds, as well as solar protection factor of ferns extracts, here's what we have. We found that the higher total phenolic compounds were at the Polypodium aureum leaves, uh, followed by the nidulary rhizomes. So those were the higher amounts of total phenolic compounds. Those that are here presented are expressed in milligrams of gallic acid equivalents per gram of dry samples. And well, as well, we have here some sun protector activity that it was not shown in all of the samples. It did not have great sun protection factor in all of them, but it did show some interesting value here, specifically in the nidulary rhizomes, the Polypodium aureum rhizomes, and as well as the S attenuatum rhizomes. So here we have some interesting data because the rhizomes are focused to have this mainly activity as a protectant factor. And and yeah, this is the main the main topic here. So where did we get with this information? Well, I can say confident to you that wild ferns have an interesting potential for skill healing formulations. Here I have that 19 out of 20 species included in this study has shown either some content of antiviral agent metabolite such as quinic and or chlorogenic acid and or antimicrobial activity. Um, Another important conclusion to this study is that most species have higher content of those metabolites in leaves. Another point here is that 10 out of 20 ferns show antimicrobial properties against gram-positive bacteria, uh, being destructive S. laudiceum and N. classifolium, the most active ones. And we found a linear correlation between the metabolite tested at the UV protection properties. Um, it was not found. So we could actually uh, say that this task effect probably is due to synergic effect between some specific polyphenols and other polyunsaturated molecules that are the one responsible for UV absorption. And one more thing that could actually be interesting to say is that gram-positive antibiotic activity um, was shown, but no gram-negative antibiotic activity was found. So it seems to be affecting the gram-positive bacteria, not the gram-negative one. And yeah, this is what we found. Some references as the methodology aspects. And I want to present to you part of the team that did this work and where we did it. We are located in Heredia, Costa Rica. We have a new building in case you want to have some uh, engagement uh, or contact. Um, this is our group because at the left of the in the photo with the people at the left here is Paulo Jimenez and at the right Victor Alvarez, and in between we have all the students here. This is our building and the team that did this work at the left. So if you have any questions at all, any comments, I would be happy to help you with those.